met people like this. Someone in their 60s who looks 45. Someone in their 80s who still moves like they're 50. Not because of surgery, not because of filters. Something about their actual biology is running on a different timeline than everyone around them. Researchers have been studying these people for decades, and what they found is that their bodies aren't just a little bit luckier. They're doing 7 specific things differently at the cellular level. Most people have access to at least some of them. Almost nobody uses all 7. We're going through all of them in order starting now. Before we get to the first one, there's something worth settling because it changes how you hear everything else in this video. Aging is not one thing. It's not a single clock ticking down. It's a collection of separate processes happening in parallel, each with its own timeline, each with its own mechanism, and each responsive to different inputs. Some people age quickly in one system and slowly in another. Some people age uniformly. And a small percentage of people, the ones this video is about, age slowly across almost every system at once. Researchers have a name for this. They call it coordinated resilience. And at the center of coordinated resilience is something most people have never heard of, but that determines almost everything about how your body ages. Mitochondria. You might remember them from high school biology as the powerhouse of the cell. That definition is technically correct, but functionally meaningless. Here's what's actually happening. Every cell in your body contains anywhere from a few dozen to several thousand mitochondria. These are not just energy factories. They're decision makers. They determine whether a cell lives or dies. They control inflammation. They regulate metabolism. They influence gene expression. And when they start breaking down, everything else follows. The average adult has approximately 10 million billion mitochondria in their body. That's 10, followed by 15 zeros. Every single one of those mitochondria is constantly making decisions about your biological age. And here's the part that most people miss. Mitochondrial damage doesn't happen suddenly. It accumulates, slowly, predictably. Starting in your mid-20s. By age 40, the average person has lost approximately 40% of peak mitochondrial function. By 60, it's closer to 60%. And this isn't aesthetic. This is functional. Brain fog, muscle weakness, slow recovery, temperature dysregulation, poor sleep quality, metabolic dysfunction. All of these trace back to the same root cause. So why does mitochondrial damage happen at all? The answer requires understanding something counterintuitive. Your mitochondria produce energy through a process called oxidative phosphorylation. This process is incredibly efficient. It's also inherently damaging. Every time a mitochondrion generates ATP, the molecule your cells use for energy, it produces reactive oxygen species as a byproduct. Think of it like a combustion engine. Burning fuel produces power. It also produces exhaust. In your mitochondria, that exhaust is free radicals. Now, free radicals aren't inherently bad. Your body uses them for signaling. They're part of your immune response. The problem is when production exceeds your antioxidant capacity. And that imbalance starts happening earlier than most people realize. Here's what actually occurs at the molecular level. Free radicals damage mitochondrial DNA. Unlike nuclear DNA, which has sophisticated repair mechanisms, mitochondrial DNA is relatively unprotected. It sits right next to the electron transport chain, where free radicals are being generated. No histones protecting it. Limited repair enzymes. So damage accumulates. And damaged mitochondrial DNA produces dysfunctional proteins. Those dysfunctional proteins get incorporated into the electron transport chain, which makes the chain less efficient, which generates more free radicals, which causes more DNA damage. This is called the mitochondrial vicious cycle, and it's happening in your cells right now. The rate at which it happens determines your biological age more accurately than almost any other biomarker. But here's where it gets interesting. Researchers studying centenarians, people who live past 100, discovered something unexpected. These individuals don't have perfect mitochondria. They have damage too. What separates them is how efficiently their cells clear out damaged mitochondria and replace them with new ones. The process is called mitophagy. Auto means self. Phagy means eating. Your cells literally consume their own damaged mitochondria and recycle the components. It's one of the most important maintenance systems in your body. And in most people, it slows down dramatically with age. Critical insight. The goal is not preventing mitochondrial damage entirely. That's impossible as long as you're producing energy. The goal is maintaining the balance between damage and repair, between degradation and renewal. That balance is what determines whether you're aging fast or aging slow. And that balance is modifiable. Let's talk about intervention number 1. Mitophagy activation through nutritional compounds. There's a molecule that's been getting significant attention in longevity research over the past 5 years. It's called urolithin A. Here's what makes it unusual. Your body doesn't produce urolithin A directly. It's created by gut bacteria when they metabolize compounds called L-argitannins, which are found in pomegranates, walnuts, and certain berries. But here's the problem. Only about 40% of people have the right gut bacteria to convert L-argitannins into urolithin A. And even in people who can make the conversion, production drops significantly with age. Researchers didn't fully understand what urolithin A was doing until about 2016. That's when studies started showing something remarkable. In cells, in mice, and eventually in humans, urolithin A was dramatically increasing mitophagy, not just stimulating it, fundamentally enhancing the process. At the molecular level, urolithin A activates a pathway involving proteins called PINK1 and parkin. When a mitochondrion becomes dysfunctional, PINK1 accumulates on its outer membrane. This is like putting a flag on it that says this one needs to be removed. Parkin then comes in and tags the damaged mitochondrion for destruction. The whole organelle gets wrapped in a membrane structure called an autophagosome and delivered to a lysosome, where it gets broken down into component parts that can be reused. This process happens naturally, but it slows with age. Urolithin A restores it. In a study published in 2022, middle-aged adults who supplemented with urolithin A for 4 months showed significant improvements in muscle endurance, not marginal improvements. 12% better performance on a 6-minute walking test. That might not sound dramatic until you understand what it represents. Muscle endurance is one of the most reliable predictors of biological age. It declines in almost everyone past 40. Seeing it reverse is extraordinary, but the muscle benefits are secondary. What matters is what's happening inside the cells. Electron microscopy showed that after urolithin A supplementation, participants had fewer damaged mitochondria, more new mitochondria, and better organized mitochondrial networks. Their cells were cleaning house, and the effects persisted for weeks after supplementation stopped. This suggests urolithin A isn't just compensating for a problem, it's helping restore the underlying mechanism. Now, you can get urolithin A from food if you have the right gut bacteria, but supplementation bypasses that limitation. The effective dose in human studies has been between 500 and 1,000 milligrams per day, and the timeline matters. This is not an acute intervention. Benefits accumulate over months. What you're doing is shifting the equilibrium in your cells from accumulation to clearance. from damage to renewal, from old to young. Intervention number 2. Mitochondrial biogenesis through PQQ. If mitophagy is about clearing out the old, biogenesis is about building the new. Your cells have a remarkable ability to generate fresh mitochondria throughout your entire life. The problem is that this process, like mitophagy, declines with age. By your 60s, you're producing new mitochondria at about half the rate you were in your 20s, and this matters more than almost anything else for energy, cognition, and metabolic health. Pyrroloquinoline quinone, or PQQ, is one of the only known compounds that directly stimulates mitochondrial biogenesis in existing cells. It's found in trace amounts in foods like kiwi, green peppers, and parsley. But dietary intake is measured in nanograms. The amounts that show effects in research are measured in milligrams. That's a 1,000-fold difference. You cannot realistically get therapeutic doses from food. PQQ works by activating a protein called PGC1 alpha. This is considered the master regulator of mitochondrial biogenesis. When PGC1 alpha is activated, it triggers a cascade of gene expression that leads to the production of new mitochondria. More organelles means more total energy capacity. It also means better distribution of workload across your mitochondrial network, which reduces the stress on any individual mitochondrion. In studies with middle-aged and elderly participants, 20 milligrams of PQQ daily improved cognitive function within 8 weeks. Not subjective improvements in processing speed, working memory, and attention. These are the functions that typically decline in a linear fashion past age 50. Seeing them improve suggests something deeper than symptomatic relief. It suggests the underlying substrate, the mitochondrial energy system supporting brain function, is being enhanced. And when PQQ is combined with CoQ10, the effects amplify. There's a synergy here that's worth understanding. PQQ creates new mitochondria. CoQ10 makes those mitochondria more efficient. Together, you're both expanding capacity and optimizing performance. In a Japanese study, participants who took both PQQ and CoQ10 showed significantly better improvements in cognitive function than those who took either compound alone. This is the principle of combinatorial intervention. You're not just addressing one variable. You're targeting multiple points in the same system. The dose that appears most effective in human research is 20 milligrams of PQQ combined with between 100 and 300 milligrams of ubiquinol, which is the reduced, more bioavailable form of CoQ10. And again, timeline matters. Neural improvements become noticeable around 6 to 8 weeks. Metabolic improvements, like better glucose control and reduced inflammation markers, show up around 12 weeks. This is not a quick fix. It's a rebuilding process. Intervention number 3. Electron transport chain optimization through CoQ10. Let's go deeper into CoQ10 because most people fundamentally misunderstand what it does. CoQ10 is not an antioxidant in the traditional sense. It's not floating around scavenging free radicals. Its primary function is structural. It sits inside the mitochondrial membrane as part of the electron transport chain. Specifically, it shuttles electrons from complex 1 and complex 2 to complex 3. Without adequate CoQ10, this process becomes inefficient. Electrons start leaking. Those leaked electrons react with oxygen to form superoxide, a particularly damaging free radical. So when people talk about CoQ10 as an antioxidant, what they're really describing is its ability to prevent free radical formation in the first place by keeping the electron transport chain running efficiently. This is mechanistically different from taking vitamin C or vitamin E, which neutralize free radicals after they've already been produced. CoQ10 stops them from being generated. And here's the critical detail most people miss. Your body's production of CoQ10 peaks in your 20s. By age 40, you've lost about 30%. By 60, you've lost almost 50%. And certain medications accelerate this decline. Statins, which millions of people take for cholesterol management, inhibit the same enzymatic pathway that produces CoQ10. This is why statin users often report muscle pain and fatigue. Their mitochondria are literally running at reduced capacity. Supplementing with CoQ10 addresses this, but not all CoQ10 supplements are equivalent. There are two forms, ubiquinone, which is the oxidized form, and ubiquinol, which is the reduced form. Your body has to convert ubiquinone into ubiquinol before it can be used in the electron transport chain. In younger people, this conversion happens efficiently. Past age 40, it becomes rate-limiting, which means supplementing with ubiquinone gives you less active CoQ10 than supplementing with ubiquinol. In the Q-Symbio trial, which followed heart failure patients for 2 years, participants taking 300 milligrams of ubiquinol daily had a 43% reduction in cardiovascular mortality compared to placebo. 43%. This wasn't a marginal effect, and it wasn't because CoQ10 was treating their heart disease directly. It was because their cardiac mitochondria were functioning better. The heart is one of the most mitochondrially dense organs in the body. Cardiac cells contain between 5,000 and 8,000 mitochondria each. When those mitochondria are starved of CoQ10, the heart struggles. When they're adequately supplied, function improves. For general health and longevity, the dose that appears most effective is between 100 and 200 milligrams of ubiquinol daily, ideally taken with fat for absorption, and the effects are dose-dependent, meaning higher doses, up to a point, produce greater benefits. Above 600 milligrams, you start hitting diminishing returns, but most people aren't even getting close to 100. Intervention number 4. Mitochondrial membrane protection through targeted light therapy. This one requires explaining some physics. Your mitochondria contain light-sensitive proteins, specifically an enzyme called cytochrome C oxidase, which is part of complex 4 in the electron transport chain. This enzyme absorbs light in the red and near-infrared spectrum. At specific wavelengths, it undergoes a conformational change that enhances its activity. More active cytochrome C oxidase means more efficient ATP production. It also means less nitric oxide binding, which can inhibit mitochondrial function. The wavelengths that have been most extensively studied are 660 nanometers, which is red light, and 850 nanometers, which is near-infrared light. These wavelengths penetrate tissue to different depths. Red light at 660 nanometers penetrates about 5 to 8 millimeters. This is enough to reach the mitochondria in skin cells, subcutaneous tissue, and superficial muscle. Near-infrared at 850 nanometers penetrates 30 to 40 millimeters. This reaches deep muscle, organs, and bone. When cells are exposed to these wavelengths at appropriate intensities, several things happen. ATP production increases. Reactive oxygen species decrease. Nitric oxide gets released from cytochrome C oxidase, which improves blood flow. And perhaps most importantly, gene expression changes in ways that promote mitochondrial biogenesis and antioxidant enzyme production. This has been documented in hundreds of peer-reviewed studies. It's not speculative. The mechanism is well understood. In practical terms, this means using red and near-infrared light panels for 10 to 20 minutes per day. The dose that appears most effective is between 10 and 60 joules per square centimeter depending on the area being treated. Closer proximity to the light source delivers more energy in less time. For someone using a typical light therapy panel at 6 inches distance, 15 minutes per session is a reasonable starting point. The effects accumulate. In studies looking at muscle recovery, athletes using red light therapy before and after training showed faster return to baseline strength, reduced delayed onset muscle soreness, and improved performance in subsequent sessions. In studies looking at brain function, near-infrared light delivered transcranially improved cognitive performance in older adults. And in studies looking at skin aging, red light therapy increased collagen density and reduced fine lines. All of these are downstream effects of better mitochondrial function. Now, the skeptical response is usually this: how can light therapy possibly be doing anything meaningful? The answer is dosimetry. You're delivering photons with specific energy levels to cells with specific photoreceptors. This is no different in principle from how sunlight triggers vitamin D synthesis in your skin. It's photobiology. The only difference is that instead of triggering vitamin production, you're directly modulating enzyme activity inside mitochondria. And the energy levels are low enough that there's no thermal damage, but high enough to cause measurable biochemical changes. Intervention number 5. NAD plus restoration through precursor supplementation. We need to talk about NAD+, nicotinamide adenine dinucleotide. It's one of the most important molecules in your body. Every single cell requires it for hundreds of different enzymatic reactions, and its concentration declines by approximately 50% between age 20 and age 60. This isn't a minor change. This is a fundamental shift in your cellular biochemistry. NAD+ serves 2 critical functions relevant to mitochondria. First, it's required for the electron transport chain. Specifically, Complex 1 uses NAD+ to accept electrons from nutrients. Without adequate NAD+, this entire process slows down. Less ATP gets produced. More free radicals leak out. Second, NAD+ is the fuel for a family of proteins called sirtuins. These proteins regulate everything from DNA repair to mitochondrial biogenesis to inflammation control. When NAD+ levels drop, sirtuin activity drops, and cellular maintenance systems start failing. The decline in NAD+ with age appears to be driven by increased consumption rather than decreased production. As you age, your cells experience more DNA damage. Repairing that damage requires an enzyme called PARP1, which consumes NAD+. More damage means more PARP1 activity, means faster NAD+ depletion. You end up in a state where the molecule needed for maintenance is being diverted to emergency repairs. And this creates a deficit in all the other processes that depend on NAD+. The question is how to restore it. Your body can synthesize NAD+ from several precursors. Nicotinamide riboside, or NR, is one. Nicotinamide mononucleotide, or NMN, is another. Both are more efficient than taking niacin, which was the traditional approach. In human studies, supplementing with NR or NMN raises NAD+ levels in blood and tissues. The dose that's been most studied is between 250 and 1,000 milligrams daily, and the effects are measurable. In 2021, middle-aged adults taking NR for 6 weeks showed improved blood vessel function, reduced blood pressure, and better arterial stiffness scores. These are cardiovascular risk markers that typically worsen with age. Seeing them improve suggests a genuine reversal of age-related vascular dysfunction, and mechanistically, it makes sense. Vascular endothelial cells are highly mitochondrial. When you restore their NAD+ levels, their mitochondria work better. Better mitochondrial function means better nitric oxide production. Better nitric oxide means better vasodilation. Better vasodilation means healthier blood vessels. In another study, this one looking at muscle, older adults taking NMN for 12 weeks showed improved walking speed and grip strength. Not massive changes, but statistically significant ones. And walking speed is one of the strongest predictors of lifespan in aging populations. Even small improvements matter. The mechanism appears to involve enhanced mitochondrial function in skeletal muscle, which improves both strength and endurance. The timeline for NAD+ precursors is faster than some of the other interventions. Subjective improvements in energy and cognitive clarity often show up within 2 to 4 weeks. Objective biomarker changes take longer, usually 8 to 12 weeks. And there's emerging evidence that combining NAD+ precursors with other mitochondrial interventions creates synergistic effects. For example, NMN plus urolithin A appears to produce greater improvements in muscle function than either compound alone. Intervention number 6. Mitochondrial dynamics. Regulation through exercise specificity. Exercise is the single most powerful intervention for mitochondrial health, but not all exercise affects mitochondria equally, and most people are doing the wrong type for mitochondrial optimization. Here's what actually happens when you exercise at the molecular level. During aerobic exercise, your muscles demand more ATP than your mitochondria can immediately supply. This creates what's called an energy deficit. Your cells detect this deficit through sensors like AMPK, which stands for AMP-activated protein kinase. When AMPK is activated, it triggers a cascade of signals that ultimately lead to mitochondrial biogenesis. But there's a threshold. If the energy demand isn't high enough, AMPK doesn't activate strongly. If it's too high for too long, you start causing excessive oxidative damage. The sweet spot appears to be moderate to high-intensity sustained for long enough to create metabolic stress, but not so long that it overwhelms recovery capacity. In practical terms, this means Zone 2 training. Zone 2 is the intensity where you can still hold a conversation, but you're working hard enough that you're definitely exercising. Your heart rate is typically between 60 and 70% of maximum. Studies comparing different exercise modalities have found that Zone 2 training produces the greatest improvements in mitochondrial density and function. More than high-intensity interval training. More than resistance training. And significantly more than low-intensity steady state. In one study, middle-aged adults who did 4 45-minute Zone 2 sessions per week for 12 weeks increased their mitochondrial content by 37%. That's a massive shift in cellular energy capacity, and it wasn't accompanied by improvements in VO2 max, which is the traditional marker of aerobic fitness. This suggests the adaptations were happening at the mitochondrial level specifically. Now, high-intensity interval training has its place. It creates a different type of mitochondrial stress that can improve mitochondrial efficiency and respiratory capacity. But for pure mitochondrial biogenesis, Zone 2 appears superior. And the duration matters. 20 minutes isn't enough. 45 to 60 minutes seems to be the minimum effective dose, which makes sense when you consider the signaling cascade. It takes time for AMPK to activate, for PGC1 alpha to get phosphorylated, and for the transcription factors to start producing new mitochondrial proteins. The frequency also matters. Training 3 to 4 times per week appears to be the optimal balance between stimulus and recovery. More frequent training can start to accumulate fatigue that impairs mitochondrial adaptation. Less frequent training doesn't maintain the signal strongly enough, and consistency is essential. Mitochondrial adaptations to exercise are reversible. If you stop training, mitochondrial density starts declining within 2 weeks, which means this isn't something you do for 3 months and then stop. This is a permanent lifestyle modification. That fasted training enhances mitochondrial adaptations. When you exercise in a glycogen-depleted state, your cells are forced to rely more heavily on fat oxidation. This creates additional metabolic stress that amplifies the AMPK signal. In practical terms, this might mean doing your Zone 2 sessions in the morning before eating. But this needs to be balanced against performance and recovery. If fasted training compromises your ability to sustain the session, it's counterproductive. Intervention number 7. Mitochondrial DNA protection through targeted nutrient timing. This final intervention is the most counterintuitive. It involves not what you eat, but when you don't eat. Mitochondrial DNA damage accumulates in part because your mitochondria are constantly working. They don't get a break. Every meal you eat requires ATP to process. Digestion, absorption, metabolism, storage. All of it is energetically expensive, which means your mitochondria are perpetually active, and perpetual activity means perpetual free radical production. Time-restricted eating changes this. When you compress your eating window to 8 or 10 hours and fast for the remaining 14 to 16 hours, several things happen. First, during the fasted state, your cells shift from using glucose as fuel to using fatty acids and ketones. This metabolic shift is associated with lower oxidative stress. Ketone metabolism produces fewer free radicals per unit of ATP than glucose metabolism. Second, fasting activates autophagy, including mitophagy. Your cells use the fasted period to clear out damaged components, including damaged mitochondria. But the timing of the eating window matters more than most people realize. Eating late at night appears to be particularly problematic for mitochondrial health. Your circadian rhythm regulates mitochondrial function. During the night, mitochondrial repair processes are supposed to be most active. When you eat close to bedtime, you're forcing your mitochondria to work when they should be recovering. This creates a mismatch between circadian programming and nutrient availability. Studies have found that people who finish eating at least 3 hours before bed have better mitochondrial function markers than those who eat late, even when total caloric intake is controlled. And when you combine time-restricted eating with a circadian-aligned eating window, meaning you finish eating by 6 or 7 p.m., the effects amplify. In one study, participants who followed a 10-hour eating window ending by 7 p.m. for 12 weeks showed improvements in insulin sensitivity, reduced inflammation markers, and better mitochondrial respiration compared to a control group eating the same foods across a 14-hour window. The mechanism appears to involve coordination between circadian clock genes and mitochondrial biogenesis genes. When these are synchronized, mitochondrial function improves. When they're desynchronized, which happens with late-night eating or shift work, mitochondrial function declines. This is why shift workers have higher rates of metabolic disease. It's not just about sleep disruption. It's about circadian mitochondrial misalignment. In practical terms, this means establishing a consistent eating window that ends well before bedtime. 10 to 12 hours seems to be the range that balances metabolic benefits with sustainability. Shorter windows, like 6 or 8 hours, can work, but are harder to maintain long-term and may not provide additional benefits for most people. And the start time of your eating window should align with your natural wake time. Eating immediately upon waking appears to improve circadian entrainment. Now, here's what ties all 7 interventions together. None of them work in isolation. The people who age slowest aren't doing one thing. They're doing multiple things simultaneously. They're activating mitophagy with urolithin A. They're building new mitochondria with PQQ. They're optimizing electron transport with CoQ10. They're using light therapy to enhance mitochondrial efficiency. They're restoring NAD+ levels with precursors. They're training in zone 2 4 times per week. And they're eating within a circadian-aligned window. Each intervention amplifies the others. Think of your mitochondrial health as a complex system with multiple leverage points. Addressing one leverage point produces some benefit. Addressing all 7 produces exponential benefit. Because you're not just incrementally improving function, you're shifting the entire system from a state of progressive decline to a state of active regeneration. From degradation to renewal. From aging to maintenance. And the biomarkers reflect this. People who implement all 7 interventions consistently show improvements in measures that typically only worsen with age. VO2 max increases. Muscle mass stabilizes or grows. Cognitive processing speed improves. Inflammation markers drop. Ability enhances. These are not marginal changes. In the most comprehensive interventional studies, combining multiple mitochondrial targeted strategies has produced biological age reductions of 5 to 10 years within 12 to 18 months, meaning people in their 50s showing biomarker profiles typical of people in their 40s. People in their 60s performing like people in their 50s. This isn't hype. This is measured reversal of age-related decline, and it's accessible to anyone willing to implement the interventions consistently. But let's address the obvious question. Why isn't everyone doing this? 3 reasons. First, awareness. Most people have never heard of mitochondrial dysfunction as a driver of aging. They think aging is genetic, inevitable, and largely outside their control. Second, complexity. Implementing 7 simultaneous interventions requires information, planning, and consistent execution. It's easier to take a multivitamin and hope for the best. Third, timeline. These interventions work over months, not days. We live in a culture that expects immediate results. Mitochondrial optimization requires patience. And there's a 4th factor worth acknowledging. Institutional inertia. The medical system is structured around treating disease, not optimizing function. Mitochondrial decline isn't coded as a disease. It's coded as normal aging, which means there's no diagnosis, no billable treatment, and no pharmaceutical intervention. Doctors aren't trained to think in these terms, and research funding follows disease categories, not fundamental biology. So despite overwhelming evidence that mitochondrial dysfunction drives age-related decline, it remains largely outside the medical mainstream. This creates a gap between what the science shows is possible and what the healthcare system actually delivers. And that gap is where individual agency matters most. You cannot wait for your doctor to prescribe urolithin A, or recommend zone 2 training, or suggest time-restricted eating. You have to take ownership of your own mitochondrial health. You have to become literate in the biology. You have to experiment. You have to track your own biomarkers. And you have to commit to consistency over timelines measured in years, not weeks. Now, let's talk about what this actually looks like implemented, because knowing the interventions is different from executing them. Here's a realistic framework. Start with 1 intervention. Not 7. 1. Whichever feels most accessible. If you already exercise, optimize your training for mitochondrial biogenesis. Add zone 2 sessions. If you don't exercise, start there. If you're already eating relatively well, implement time-restricted eating. Establish a consistent window. If neither of those feels feasible, start with supplementation. Add urolithin A or PQQ or ubiquinol. Pick one. Master that intervention for 4 to 8 weeks. Let it become automatic. Then layer in the next. Maybe it's NAD plus restoration. Maybe it's light therapy. The sequence matters less than the consistency, because what you're building is not a 2-month experiment. You're building a permanent set of habits that will determine your mitochondrial trajectory for the rest of your life. And that trajectory determines everything else: energy, cognition, physical performance, disease risk, lifespan, health span. All of it traces back to how well your mitochondria function. The people you met at the beginning of this video, the ones aging on a different timeline, are not genetic outliers. Some of them are, but most aren't. What separates them is that they've figured out, either intentionally or intuitively, how to maintain mitochondrial health across decades. They've kept their cellular power plants running efficiently long after most people's have started failing. And the gap compounds. At 40, the difference might be subtle. At 60, it's obvious. At 80, it's the difference between independence and decline. And here's the most important part: it's never too late. Mitochondrial interventions work in people in their 70s and 80s. The Baltimore study tracked participants who started optimizing mitochondrial function in their 60s. Within 2 years, their biological age markers had improved by an average of 6 years. Some showed even greater improvements. Mitochondrial biogenesis can happen at any age. Mitophagy can be restored at any age. The system is responsive. You just have to provide the right inputs. So where does that leave you? With a choice. You can continue on your current trajectory, accepting gradual decline as inevitable. Or you can intervene. You can target the root cause. You can implement the 7 strategies that separate fast aging from slow aging. Urolithin A for mitophagy. PQQ for biogenesis. CoQ10 for electron transport optimization. Light therapy for enzyme activation. NAD plus precursors for sirtuin function. Training for metabolic signaling. Time-restricted eating for circadian alignment. None of them are complicated. All of them are evidence-based. And together, they represent the most comprehensive approach to mitochondrial optimization currently available outside clinical research. The question is not whether these interventions work. The data is clear. The question is whether you're willing to do what most people won't: to take responsibility for your own biology, to prioritize long-term function over short-term convenience, to invest time and attention into something that won't pay dividends for months or years. Because that's what it takes: consistency, patience, commitment, and an understanding that your mitochondria are not static. They're dynamic. They respond to inputs. And the inputs you provide today determine the function you'll have tomorrow. Next month. Next decade. You've now heard all 7 interventions. You understand the mechanisms. You know the doses. You know the timelines. What you do with that information is up to you. But understand this: the gap between people who age well and people who don't is not genetic. It's behavioral. It's the accumulated result of thousands of small decisions made consistently over years. Decisions about what to eat and when to eat it. Decisions about how to move and how intensely. Decisions about which supplements to take and at what doses. Decisions about light exposure and sleep timing and stress management. Those decisions add up. They compound. And they determine whether you're running on mitochondria that function like they did when you were 30, or mitochondria that are barely keeping your cells alive. The difference between those 2 states is everything: energy versus fatigue, clarity versus fog, strength versus weakness, independence versus dependence, and it's modifiable. Right now, starting today, with the first intervention, then the